Welcome back to my channel. My name is Dr. Anwar Youssef Dunbar, and this is Big Discussions 76, Science and Technology. First of all, please like this video, please share it, and please subscribe to my channel. Well, I actually have a couple of things in the works for this channel in terms of content that I want to upload. I'm almost done remastering my pharmacy video, and I have a fun interview with a peer from the aerospace sector that I want that I'm working on right now in terms of out processing and editing and stuff like that. But the coronavirus and chloroquine and hydroxychloroquine has jumped back to the top of the pile. And I think this will be the first time on this channel that I'm actually presenting something from the scientific literature. So in this offering, I'm going to show you some data and walk you through the data and discuss what it means, just like my buddy Nicole Ali. Nicole, if you look at this, please weigh in and give your expertise on the data that I show. I'm pretty outspoken about my expertise as being in pharmacology and toxicology, and those are sciences that basically look at what exogenous or endogenous molecules do to living systems when those systems are exposed. So pharmacologists and toxicologists are masters, excuse me, of what molecules do to living systems. And there is some cell biology and virology and immunology and parasitology involved, depending upon which part of pharmacology you go into. I did take some of our core courses at the University of Michigan did involve some protein structure and function and molecular and cellular biology. But one thing as a scientist is that sometimes you have to brush up on those topics and read up on your basics and refresh on your basics. So in any case, I was on a panel recently discussing the 2020 election and misinformation, and of course the coronavirus came up, and the fact that the Trump administration endorsed hydroxychloroquine back in March came up, and the fact that there were a group of doctors who stated that particularly an African doctor stated that it worked and she was able to see efficacy in her patients. It came up on this panel that video was suppressed and that information was suppressed. And I want to acknowledge Sir Tice for sending me that video and allowing me to look at it and look at the statement by this group of doctors and this particular doctor from Africa. If I can leave that video in the description box below this video, I will. I don't, we're in an era where, you can't say certain things, and certain things I believe are intentionally suppressed. So, I'm going to give that some thought in terms of leaving that link in the description box below. But I'm saying all this to say, and Sir Tice sent me that video because there's uncertainty regarding whether or not hydroxychloroquine actually works. And I've recorded several videos on hydroxychloroquine and chloroquine right here on Big Discussions 76, Science and Technology. It's been a learning experience, and I actually found out through asking questions about whether or not those drugs are effective for COVID-19, I actually found that there's another concern for the use of those drugs, and that is for individuals with glucose 6-phosphate dehydrogenase deficiency. So there's a whole community of people who are susceptible to hemolytic anemia if treated with hydroxychloroquine. I think that's an important part of the story. So with that acknowledged, the residual question in my mind is for people not deficient in G6PD, is this a viable strategy and treatment for COVID-19? And there's uncertainty about whether that's true. Now, I did, back in March, when all of this was unfolding, the virus, potential treatments, the spiking number of cases, and the inaccurate testing, I started doing some digging to kind of familiarize myself on this topic, and I found an article this is dated March 18th, 2020. So this is about 5 months old. Let's see, it's August, March. Yeah, this is about 5 months old. And so we know that the virus has mutated. Nicole has talked extensively about that. But what I want to address is whether or not hydroxychloroquine and chloroquine actually work in terms of inhibiting the virus. And so this article is entitled Hydroxychloroquine, a less toxic derivative of chloroquine, is effective in inhibiting SARS-CoV-2 infection in vitro. So I'm going to try to walk through this at a very high level so that the non-scientists can take something from it, and just so that it's understandable for a general audience. I don't want to read this thing word for word, but I'm just going to try to walk through it at a high level, and so that people can glean from it the important points. And it looks like, looking at the authors, this was done by an Asian group. And this is from the journal Cell Discovery. And I don't know if any reports have come out after this. And so in terms of pharmacology and toxicology, ultimately we're interested in the effects in the whole organism. But oftentimes the first step is conducting experiments in vitro. So down at the very protein level. So just looking at how the drug perhaps impacts the activity of an enzyme. Does it inhibit the enzyme? Does it block its function? Or does it modulate the enzyme in such a way that increases its function? So when you're looking at what drugs do, you want to characterize those very, very early events to see what it does at the protein level. Also, you could, in terms of DNA and RNA, does it bind nucleic acids? And then from there, you go to the cell and you want to try to map out what it does on the cellular level. If it binds DNA, what does that do to the downstream cascades within the cell? Does it block the production of a hormone? Does it blockade a receptor? Does it go into the membrane of the cell and modulate that receptor on the inside, or modify that receptor on the inside such that the downstream signaling pathways are blocked or enhanced? Does it inhibit the enzyme of focus? So again, you want to map out the early events, and those give you ideas as to what will happen on the tissue and the organ level, and what will ultimately happen to the whole organism. And that's what this paper is looking at. So at this time, when this was submitted, remdesivir, and I've uploaded content on this channel about remdesivir. That's an antiviral. That was in the early stages of development. They talk about that here in the opening paragraph. Let's see. Chloroquine has long been used to treat malaria and amoebiasis. I'm not sure what amoebiasis is, but I think it's probably related to malaria. Here they're mentioning the plasmodium, and anyone who's taken a zoology class knows that malaria is caused by the plasmodium and not the mosquito. Shout out to Dr. Thompson, who was one of my professors at Johnson C. Smith University. He quizzed us on that all the time. It's not the mosquito, it's the plasmodium that rides along inside the mosquito and goes into our bloodstream when the mosquito tries to feed. Let's see. So chloroquine has been used to treat plasmodium. Hydroxychloroquine sulfate is a derivative of chloroquine. Was first synthesized in 1946 by introducing a hydroxyl group onto the chloroquine. Let's see. It's been demonstrated to be much less toxic than chloroquine in animals. And hydroxychloroquine is still widely available to treat autoimmune diseases such as systemic lupus, erythematosus, and rheumatoid arthritis. So these drugs have been around for a while, and they are well characterized. And I said that in one of my videos about hydroxychloroquine. So the question is whether or not this has the ability to attenuate, which is a word for decreasing or minimize. Does this have the ability to attenuate the ability of the coronavirus to infect living systems? So what they did, just briefly, in the study is they used monkey kidney Vero E6 cells. So usually in these types of experiments, you want to select a mammalian cell line that would serve as an adequate model for human cells. And they may have gone forth after this paper to do experiments in actual human cell lines, but this was in monkey kidney Vero E6 cells. And so what they did is they performed a cytotoxicity assay with hydroxychloroquine and chloroquine. So what you want to do when you do these types of studies is you want to make sure that the drug of choice does not kill the cells. So you want to make sure that the cells can tolerate the drug. So they performed initial cytotoxicity studies, and then they performed a series of dose response studies, under varying multiplicities of infection of those cell lines. So I'm going to jump us to, and this is the only figure in this paper. So looking at figure 1, this is entitled Comparative Antiviral Efficacy and Mechanism of Action of Chloroquine and Hydroxychloroquine Against SARS-CoV-2 Infection in Vitro. So this is in cell lines. So if you look at figure 1A, this first plot is looking at percent cytotoxicity. Okay, so they're looking to establish a dose or concentrations, because in cells you're using concentrations. And so if you look at the x-axis for all of these plots, the x-axis depicts micromolar, and molar, micromolar, nanomolar, picomolar. Those are all designating concentration. So for example, if you look at this plot here, looking at cytotoxicity, you're going from 3.7 micromolar up to 400 micromolar. So you're increasing, you're giving the cells an increased concentration. As you can see, as you increase the concentration, you're increasing the cytotoxicity. So there's only so much of these drugs that these cells can tolerate. So they chose a testable range, which was most likely spanning somewhere in the 3 to 33.3 micromolar range. And so when you look at the other panels, you see MOI. MOI stands for multiplicity of infection. And so what that is basically they gave these cell cultures increasing amounts of the virus. So they wanted to see if, with increasing amounts of the virus, how chloroquine and hydroxychloroquine fared in terms of inhibition of the viral multiplicity. So as scientists, you want to look at the x-axis. That's usually your independent variable, which means that you're changing something. In this case, the concentration of the drug, and then you have your dependent variable. So that's the thing that's going to change based upon your independent variable. So we're looking at increased drug concentration, and we're looking at percent inhibition of the viral multiplicity. And again, this is called a dose response. This is critical to pharmacology and toxicology. So I'm going to blow this up here for myself. And what you're seeing here with the multiplicity of infection of 0.01 is you're seeing that as you increase the concentration of the chloroquine and the hydroxychloroquine, you're seeing for both that the inhibition goes up in a dose responsive manner. So I'm going to introduce some terms here. So if you see here, for each plot, there is a EC50. And so for chloroquine, for this MOI of 0.01, the EC50, the effective concentration 50. So that's the concentration at which there's 50% inhibition of the multiplicity of infection. And for toxicology, you have, for acute studies, you have an LC50, an LD50. So that's the lethal dose, which kills 50% of the animals. But in this case, the EC50 for chloroquine is 2.71 micromolar, and for hydroxychloroquine, the EC50 is 4.51 micromolar. So the chloroquine is slightly more potent than the hydroxychloroquine. But as a scientist, 2.71 and 4.51, those are not very different. SI, can I quickly see what SI stands for? I think that is looking at the legend here. Okay, so in terms of SI, the International System of Units, abbreviated SI from the Système International, the French version of the name, is a scientific method of expressing the magnitudes or quantities of important natural phenomena. There are 7 base units in the system from which other units are derived. Okay, so going back to our data, for the multiplicity of infection of 0.02, so they're increasing the viral titer. What you see is that for chloroquine, the EC50 is 3.81 micromolar, and the hydroxychloroquine has an EC50 of 4.06. So those are pretty similar. For the multiplicity of infection of 0.2, so they increased it tenfold. So again, we're increasing the concentration, basically the concentration of the virus, or the ability of the virus to infect the cell lines. So for the MOI of 0.2 for chloroquine, the EC50 is 7.14 micromolar, and for hydroxychloroquine, the EC50 is 17.31 micromolar. So when you increase the viral load to this amount, for these sets of experiments, the chloroquine is twofold more potent or efficacious than the hydroxychloroquine. For an MOI of 0.8, it's interestingly so, you're increasing the multiplicity of infection fourfold. The EC50 for chloroquine is 7.36 micromolar, and the EC50 for hydroxychloroquine is 12.96 micromolar. So again, this is a twofold difference. And so with increased viral load. Yeah, and across the board, aside from the multiplicity of infection of the 0.02, that one seems to be the outlier where they're kind of right on top of each other. But for the others, the chloroquine seems to be twofold more efficacious or potent than the hydroxychloroquine. So looking at B is a quantification of what they did is they tried to track the localization of the viral particles. And so the first bar chart for B is looking at early endosomes, and they used a viral antibody, which is called, which is binding the viral nucleoprotein. I believe that's what that is. And so if you look at the bar chart, so once again we have the pastel blue, that's our untreated, and then this pink color, and then the lavender color. The pink color is the chloroquine, and what you see is that the chloroquine has caused an increased co-localization of the virus. The hydroxychloroquine, represented by the lavender bar, is causing that effect as well, but to a lesser extent than the chloroquine itself. So as a scientist, and again, I'm not a cell biologist, though I had taken some coursework in that, these drugs are causing a difference, and they're causing the virus, it seems, to localize in a specific part of the cell, or I'll say aggregate. If someone is watching this and you want to correct me on that term, feel free to go ahead. So that's looking at the early endosomes. So the set of data adjacent to it is looking at percent co-localization in endolysosomes. And I did a quick search on these. And so the reason this is significant, so the early endosomes are the primary sorting station in the endocytic pathway from which endocytosed molecules can be recycled back to the cell membrane or targeted to degradation in lysosomes. So with that definition, what this data is telling me is that these two drugs are causing the virus to be packaged for degradation or export out of the cells. And again, if I'm wrong, please correct me. The goal here is to get to the truth and not who's right per se. So in terms of the endolysosomes, where's that definition? Enveloped viruses need to fuse with a host cell membrane in order to deliver their genome into the host cell. While some viruses fuse with the plasma membrane, many viruses are endocytosed prior to fusion. So, what this data is showing is that in B, for the untreated cells, you're getting a large amount of viral particles entering the cell via these endolysosomes. And what this data is showing, and it looks pretty clear in terms of magnitude of the changes, in terms of the standard deviations and the statistical significance, which is designated by the 3 stars, is that these 2 drugs are inhibiting and preventing the virus from entering the cells via these endolysosomes. So these drugs seem to be pretty effective at inhibiting the virus from entering into this cell line. And I was looking at C, which is this is actual imaging of the cells of the antibodies that they were using to track the movement of the virus into the cells with the use of these drugs. And I was trying to, as a pharmacologist, I was trying to make sense of what I was seeing here. So you have your untreated, which is the first column. The blue, that is the nucleus. So I think they took a picture of specific cells at specific magnifications. And so if you look at the blue, those are the nuclei. And for the untreated, those are the early endosomes. So you're looking at the early endosomes on the top row, and you're looking at the endolysosomes on the lower row. And then so going across, you're looking at the chloroquine at 50 micromolar, and you're looking at the hydroxychloroquine at the 50 micromolar as well. And what you're looking at is you're looking for differences in the I'll call it the traffic pattern. I'm not sure if that's the right term to use for this, but you're looking at differences in the packaging of the viral particles and how the viral particles are situated around the nucleus and how they're situated in the cytoplasm. And based upon the bar chart, and you could see with these arrows that the drugs are causing a different pattern of aggregation and a different pattern of localization within the cells when these cell lines are infected. And according to those bar charts, again, these bar charts are generated based upon these trafficking patterns, and what this data is showing is that these drugs are impacting the percent co-localization of the viral particles. And so what we can see is that these drugs are causing an increased percent co-localization in the early endosomes and a decreased percent co-localization in the endolysosomes. So these drugs seem to have an effect on the virus's ability to infect mammalian cell lines. So I'm going to stop this here. I'm going to leave this article in the description box below. And so again, the overarching question was, in my mind, was why were they interested in this drug in the first place? Regarding the Trump administration and also the scientists that thought that this would be a viable treatment for COVID-19. And looking at this early in vitro data, the answer is right here. It seems to have an impact on the ability of the virus to get inside mammalian cells and infect them and reproduce and incorporate its own machinery. Now, going back to pharmacology and toxicology, in vitro testing is only the first step. Once you establish that your drug actually works at the protein level and in cells, then you have to go ahead and see how it, and if it works in animals at the tissue and organ level and how it affects the whole organism. And with that, there are a host of other variables, such as pharmacokinetics, absorption, distribution, metabolism, and excretion, ADME, which I plan to discuss in another video. And then you have to think about the best way to deliver the drug to the site of action, and then does it cause toxicity in some other way. So I'm going to stop this here. I hope someone was able to glean something from this whole discussion. If anyone has any questions or comments, please leave those in the comment section below. Please like this video, please share it, and please subscribe to my channel. And as always, remember that your attitude determines your altitude. Take care, and I'll talk to you the next time. Bye-bye.